Good afternoon, everyone. My name is Ajalutim Tope Angel. I'm here to discuss the biological application of phytochemistry, including vision, bioluminescence, and vitamin D synthesis. We have the introduction. Phytochemistry is a branch of chemistry that studies chemical reaction, processes, and transformation initiated or influenced by the absorption of light, typically the ultraviolet, visible, infrared, and so on and so forth. It explores how light energy in form of proton interacts with molecules to synthesize electrons, vibrational or rotational transaction, leading to chemical changes. These changes can include bond breaking, bond formation, isomerism, electron transfer, which are fundamental to various natural and synthetic processes. We have biological phytochemistry processes. Biological phytochemistry processes encompass the studies of chemical reaction and processes triggered by light in living organisms. These processes are fundamental to life, driving crucial functions such as photosynthesis, vision, and photoprotection. By harnessing light energy, biological systems, converting it into chemical energy, regulate growth and protect against damage. This write-up provides a comprehensive overview of key biological phytochemical processes, their mechanism, their significance, with a focus on the vision, vitamin D, and bioluminescence. Let us go to the photochemistry of vision. The photochemistry of vision is a fantastic interplay of light, molecular transformation, and biochemical signaling that enables organisms to perceive their environment. At the core of this process is the interaction between protons and specialized photoreceptors molecules in the retina, which covers energy light and energy into electrical signals that the brain interprets as visual information. This process primarily involves the photopigments in vertebrates and its analogues in other organisms. The phytochemistry of vision encompasses the phytoisomerism of retinal, the activities of the cardia, and the regeneration of photopigments through the visual circuits. We have the structure and function of photoreceptors. The human retina consists of two main types of photoreceptor cells, the rods and the conoids. The rods are highly sensitive to low light level and are responsible for sculptonic, which is night vision, while cones operate under daylight condition and mediate with colour vision. Both cells type contain photopigments, which are light sensitive molecule embedded in membranes embedded in the membranes of the outer segment. We have the biological application and implication of vision. We have the visual sensitivity and adaptation. The phytochemistry of vision enables remarkable sensitivity and adaptivity. Rods can detect single protons, a feature attributed to high efficiency of rhodopsin phytochemical reaction. Additionally, this visual system adapts the light condition through photochemical reaction. In bright light, photopigments are bleached, which means the retina is converted to all trans and disassociate from phonemes, reducing sensitivity to prevent saturation. In daylight, the visual circle regenerates photopigments, restoring sensitivity. This dynamic regulation underlines light and darkness adaptation. We have the photoreceptors degradation and diseases. Defects in the photochemical machinery of vision can lead to retinal disorder. Similarly, excessive light exposure can overwhelm the visual circle, leading to the accumulation of toxic retinal derivatives, contributing to age-related muscular degradation. We have the photochemistry in non-mammalian vision. Phytochemistry underpins visions in non-mammalian species, often with unique adaptation. For instance, in many vertebrates like fishes and birds, vision tends into the ultraviolet spectrum due to specialized openings. In some invertebrates such as metamorphines, a stable light source, eliminating the need for complex visual circle. These can varies highly in the evaluation flexibility of phytochemical mechanisms in vision. We have the emerging research and application. Recent advances in phytochemistry have expanded its application in vision research therapy. We have the autogenetics, a unique genetics that uses light sensitivity protons to control cellular activity, draw inspiration from the photochemistry of Rhodes and pinoids by introducing microbial pinoids like channel rod pins into retinal neurons. Which researchers have restored light sensitivity into animal modes of retinal degradation, offering potential treatments for blindness. We have some challenges in vision, which includes environmental constriction, low light conditions such as those experienced in the deep sea environment or at night limit visual authenticity. For instance, deep sea organisms often rely on alternative sensory mechanisms due to insufficient light penetration. We have physiological limitation. Age and diseases impairs vision. Age-related muscular degradation affects the retina, leading to central vision losses. We have color blindness and other genital disorders that limit visions. Clinical consideration of vision in animal and human. We have animal and human testing. Studies on vision phytochemistry frequently use animal models such as rodents or primates to understand retinal function or develop treatments for visual implementation. The use of animals raise concern about their welfare and adheres to ethical guidelines like the 3 Rs, which are replacement, reduction, and refinement, to minimize alarms. Similarly, in human clinical trials for vision, enhancing technology or gene therapy require vigorous, informed consent process to ensure participants understand risks such as potential vision loss or other side effects. We have other condition like we have under consideration like genetic editing and enhancement. Advance in genetic therapy such based on treatments for retinal diseases retinal diseases like lobia conjectitis and maros offers hope for restoring visions. We have privacy and surveillance. Phytochemistry research has led to technologies like retinal implants and bionic eyes, which could potentially be hacked or misused for surveillance purposes. We have phytochemistry of bioluminescence. Bioluminescence, this is the emission of light by living organisms in a fantastic phenomenon observed in various species, including marine organisms like jellyfish, deep sea fish, as well as terrestrial organisms such as fungi. These produce results from chemical reaction that produce light energy typically in visible spectrums without significant heat generation, a process termed cool light. The phytochemistry of luminance involves complex molecular interaction primarily centers around the oxidation of structure known as luciferin characterized by an organism enzymes lucifers. Factors influencing bioluminescence. The emission wavelengths and intensity of bioluminescence are influenced by several factors. We have the luciferin structure. The active site of the luciferin modes modulates the energy of the excited state. We have the pH and temperature. The microenvironment affects the filtration state of oxy-luciferin, altering emission color. For example, firefly luciferin emits red light at low pH due to ketocontamination of oxy-luciferin. We have cofactors. In some systems, cofactors like ATP, magnesium, calcium are essential for catalyst or triggering the reaction. Energy transfer. In organisms with accessories like energy transferred, mechanisms enhance or shift emission wavelengths as seen in aquaria. Application of bioluminescence. Bioluminescence have found excessive application in biotech and biomedical research. We have reports engine. Fly luciferin and GPS are widely used as a reporter gene to study gene expression and protein interaction in vivo. We have imaging. Bioluminescent protein enables non-invisible imaging of tumors and infection in animal modeling. We have environmental monitoring. Bioluminescent bacteria are used to detect toxins in water as their light emits decrease the presence of pollutants. We have the clinical ethical consideration. Environmental impact. The extraction of bioluminescent proteins such as luciferin or green fluorescent protein often involves harvesting organisms from their natural habitats. We have biotechnological manipulation. Bioluminescent agents are increasingly used in genetic engineering, for example, to create a glowing plant or animal for aesthetics or scientific purpose. This raises ethical questions about the commodification of life and potential for creating organisms with no clear ecological or societal benefits. We have cultural and social implication. In some culture, bioluminescent organisms hold spiritual or symbolic significance. Here are some questions about the cultural sensitivity in scientific for sports. Now we are going to the vitamin D synthesis through phytochemistry. Vitamin D is often referred to as the sunshine vitamin, is a fat soluble essential for maintaining calcium and phosphate homeostasis, supporting bone health and modulating influence functions. We have the role of UVA radiation. Vitamin D synthesis begins in the skin, specifically in the epidermal layer, where the 7 hydro hydro hydrodecholesterol, a cholesterol precursor, is in abundance. When the skin is exposed to UVA radiation, DCH, which is the 7 dehydrocholesterol, absorbs these proteins, initiating a chemical reaction. The UVA energy causes the B ring of the 7-DC to undergo a cleavage, forming pre-vitamin D. We have factors influencing vitamin D synthesis. We have skin pigments, we have season, sunscreen and clothing, we have age, atmospheric condition, and so on and so forth. We have importance of vitamin D in humans. The role of vitamin D in the human bone. One of the primary functions of vitamin D is its role in calcium absorption and bone health. Vitamin D protects the absorption of calcium in the small intestine, which is crucial for the formation and maintenance of healthy bones. Without adequate vitamin D, a small amount of dietary calcium is absorbed, leading to conditions such as rickets in children. We have the immune system support. Vitamin D is also essential for a robust immune system. Emulates both it emulates and adapts immune system response, enhancing the body ability to fight infections and reduce inflammation. Vitamin D is also used in mental health and conjunctivity function. It is used in preventing of chronic diseases. It is a source of it is also used as a source of nutrients to humans. Challenges in vitamin D. We have environmental factor, physiological barriers, lifestyle and cultural factors. Ethical consideration. We have public health intervention. We have inadequate access to alternative, and so on and so forth. Let us go to the conclusion. In conclusion, luminance and vitamin D synthesis Vitamin D synthesis. We will talk about the vision. The vision enables environment and engagement. The bioluminescence facilitates survival in light-scarce habitats, and the vitamin D synthesis supports physiological integration. Together, they reflect the adaptability of life to diverse ecological needs, and environmental and sociological changes continue to shape light availability. Thank you.
Thank you very much. Thank you very much. I am impressed. I don't know if if we actually listen to what she did here, and that was I like the flow. I like the flow. Please let's clap for her again. I like the work. I like the work. Thank you very much for the wonderful presentation. So we'll be right back for the next presentation. All right.